Good morning, good afternoon, and good evening, everyone, and welcome to this Scientist.com webinar titled Current and Novel Immuno-Oncology Drug Evaluation Methods via Humanized Mouse Models. I'm Megan Loy, the Category Director of In Vivo Services here at Scientist.com, and I'll be your host today. This webinar has been sponsored by LIDE Biotech, so a big thanks to them for helping to make this webinar possible. We're very fortunate to have Dr. Bin Shi, the Director of Immuno-Oncology at LIDE Biotech, here with us today, whose presentation includes an introduction to immuno-oncology drug development and the importance of humanized mouse models for immuno-oncology research, an overview of different humanized mouse models available for drug testing, evaluation of current IO platforms, including analysis of several case studies, and examples showing how spatiotemporal interactions between tissue-infiltrating immune cells and cancer cells can be visualized using imaging mass cytometry. Without any further ado, I'm pleased to welcome Dr. Bin Shi. Bin, thank you so much for joining us today. The floor is yours whenever you're ready.
Hello, everyone. Welcome to the webinar. I'm very pleased to give a talk about our drug evaluation via humanized mice models today. So I'm from Shanghai, China, LIDE Biotech. So this is a brief introduction of myself. I got my PhD degree from Zhejiang University, mainly focused on innate immunity. We identified two key membrane surface proteins, Siglec-G and HBDD3. We found they are important for macrophage and dendritic cell cytokine release and functional immunity. Then I moved to a University of Oxford for studying the dynamic interaction between macrophage and lymphocyte trafficking in resident tissue and discovered one novel B-cell molecular CBTB18, and we published it as a potential cancer suppressor. Before joining LIDE, I'm the research director of Shanghai GV20 Oncotherapy, and leading a big group using CRISPR screening to identify novel IO targets and support drug development. So with more than 10 years of experience in immunology, we are happy here to do some translational work in LIDE Biotech. So what is immunotherapy? Immunotherapy is one kind of cancer treatment that helps your immune system fight and beat cancer. Recently, cancer immunotherapy is becoming more and more popular. I think the earliest case of cancer immunotherapy can be tracked back to 1891, when a New York surgeon, William Corey, first attempted to leverage the immune system to treat cancer after he noticed that the mixture of live and inactivated Streptococcus bacteria could induce tumor regression in some sarcoma patients. However, Corey's toxin was not well applied in clinic since its inconsistency, and most importantly, it was fully beaten by radiotherapy and chemotherapy at that time. So nearly after 1 century. Stephen Rosberg, another US surgeon, did find that interleukin-2 treatment can successfully boost host immune system and suppress cancer development in some advanced melanoma patients. However, due to the high toxicity of systemic IL-2 treatment, such as capillary leaking syndrome, the use of interleukin-2 was also limited in clinical. So since after, immunotherapy went to a dark era, and people who did such work were under-criticized until March 2011. The first CTLA4 antibody was approved by US FDA for advanced melanoma patients. Then another famous T-cell checkpoint, PD-1, and its ligand PDL1, their antibodies were approved by FDA for melanoma, lung cancer, and brain cancer in the next few years. Now, CAR T-cell therapy was great succeed in blood cancer area by targeting CD19 or BCMA, while the first TCR-T therapy, GP100 TCR, and the CD3 bispecific fusion protein was approved by FDA for HLA-A2 positive advanced uveal melanoma patients at January this year. We believe that more and more novel immunotherapies, such as antigen-specific tumor infiltrating lymphocyte therapy, are also on the way. There is some data from Cancer Research Institute, which was founded by the daughter of William Corey. IO drugs of 2020 was more than twice as 2017, and the increased pipeline mainly focused on cell therapy or a T-cell based immunomodulate or biospecific antibodies. And the top 5 targets of 2020 were CD19, tumor-associated antigens, PDL1, PD1, and BcMA. And in the last year, more and more novel cancer therapy targets have been identified and started clinical translation, such as ID3 and SOX4. Here is very interesting. This 2 molecules was reported by Kajun Group. They reported that conventional CAR T cells underwent to NK cell-like phenotype for T cell exhaustion, while laboratory knockout either ID3 or SOX4, they delayed the T cell dysfunction by switching NK-like phenotype to effect T cell phenotype. And they did some ex vivo anti-tumor assays, and it did very well. Probably they are now testing those engineering CAR T cells on patient-derived xenograft models or IIT project patients. And another wonderful novel target is neutrophil elastase. Last year at June, Leverbeck group from Chicago University, they discovered a human, but not mouse, neutrophil release factors that selectively kill cancer cells while sparing proximal healthy cells. Further study identified was human neutrophil elastase played an important role. The most interesting part is that purified mouse neutrophil elastase can also kill both human and mouse cancer cells. However, its function was fully silenced during its secretion, as it binds with protease inhibitor called secretory leukocyte peptidase inhibitor. So it seems helpful for us to directly use humanized mouse models for IO drugs test, as it's more reflectable to human immune system. So currently, there are several humanized mouse models for IO drug evaluation. One is the CD34 hematopoietic stem cell model. The immune deficient mouse reconstitute with the stem cell. So the advantage of this model is fully human immune system with delayed or no graft-versus-host-disease. But there was no functional myeloid cells and NK cells and T cells with functional impairment, although its phenotype seems fine. And most importantly, it is expensive and time-consuming. Another is PBMC humanized mouse model. It is classic. However, due to the limited tumor infiltrating lymphocyte and common GVHD reaction, the therapeutic window of eye drug testing can be highly limited. The last model I would like to introduce is the human PBMC and the cancer cell co-inoculated model, which is maximally mimic tumor and immune microenvironment with delayed or no GVHD reaction. And in our hand, it has been successfully for various eye drugs evaluation. I divided the main topic into 4 parts. Before we talk about the humanized model, we start with syngeneic mice model and a humanized transgenic mice model, which is using mice immune system for cancer irradiation. Some antibodies not only recognize human antigen as well as mice antigen, so it's easier to run a classic syngeneic mice model for quick test. However, a lot of human antibodies only recognize human antigens, but not mouse, such as PD-1 antibody, Keytruda. So scientists engineered and replaced mouse antigen, such as the extracellular domain of mouse PD-1 with human PD-1, and constructed the cancer cell line with human PDL1 antigen for some in vivo IO drug tests. Currently, there are more than 10 different IO targets. Human transgenic mouse are commercially available. We highly recommend it, like human PD-1 and human PDL1, human CTL4 transgenic mouse. It gives a very nice in vivo efficacy. Now I will mainly introduce the PBMC humanized model here, optimized and developed in LIDE biotech. One is our donor selected classic PBMC humanized mice model. So here PBMC donors were selected and transferred to human bearing NCG mice and given drugs test for further in vivo efficacy. We know that different PBMC donors will have various T-cell reconstitution rate as well as anti-tumor effect. But using our unique PBMC donor selection method in LIDE, we can easily check in vivo efficacy of IO drugs. Here I show example of gastric cancer was well suppressed within the increased dose of anti-Claudin 18.2 antibodies.
Ben, this has been a fantastic presentation so far, but if you'll pardon the interruption, I just had a quick question here. Why was it that you used NCG mice in your PBMC humanized model? Have you tried any other strains?
True. We have tried a lot of other mouse strains, such as SCID mouse and NSG from Jackson Lab. And in our hands, we did find that NCG mouse showed a delayed or longer survival time before they happened the GVHD reaction. So that give us a longer therapeutic window for IO drug tests. That's why we mainly use NCG mouse in our facility. Yeah, thanks.
Yeah, it makes sense that you'd want a longer time frame to evaluate IO drugs before the development of GVHD. Thanks, Finn.
So here I show an example for the PD-1 antibody resistant melanoma PDX humanized model. So we did find some novel cancer target drugs seems have some effect on those special patient samples. And the T-cell based biospecific antibodies or moderate showed nice effect in ovary cancer humanized models. Yeah. as well as some colon cancer and gastric cancer models. As I mentioned before, the therapeutic window of classic PBMC humanized model is narrow as well as the limited infiltrated T cells. So in the last 10 years, we have developed a human PBMC and a cancer co-inoculate mice model. In this model, human PBMC was primed with target cancer cells, then mixed with fresh cancer cells in matrigel before co-engraftment into the NCG mouse. Drugs will be given for treatment during or after the tumor reach around like 50 to 100 cubic millimeter. So we first validate this model. The immunofluorescent images showed that the green CD3 positive T cells, they are well infiltrated in the tumor tissue. And we also did a flow cytometry analysis, which revealed that around like 3% CD3 positive T cells infiltrated in the tumor tissues at day 25 after human PBMC and cancer co-inoculating. So in this model, you will see either therapeutic anti-PD1 antibody and anti-PD or PD1 antibodies. Nearly 100% suppressed all the melanoma cell growth. In addition to PD1 and PDL1, more and more checkpoints were identified recently, such as like CD155, CD112, and bound to TIGIT, and Heaven and BCLA, VISG3 and VISTA. We used our PBMC and A375 melanoma cancer cell line co-inoculate model, and this model worked very well for anti-CD73 antibody and TIGIT antibody, as well as some GITR agonist antibodies as well. Besides our melanoma and PBMC mixed model, our lung cancer and PBMC co-transfer model works also great for DLL3 and CD3 bispecific antibodies. And PDL1 and anti-PDL1 and TGF-beta bispecific antibody work nicely in our PBMC and MDA MB231 breast cancer mixed model. Yeah. Since we had built a national biobank covering more than 1,600 PDX models in-house, particularly for those drug-resistant patient-derived tumor samples like PD1 antibody resistant. They also having some like BAF mutations and some like non-small cell lung cancer PDX models, not only to PDL1 antibody resistant, also to EGFR inhibitor resistant. So those PDX models are ready for IO drug evaluation and novel targets identification. We did find like cancer cell surface PDL1 expression was a useful biomarker for anti-PD1 therapy in vivo. Anti-PD1 antibody work very well in PDL1 highly expressed non-small cell lung cancer patients, but not in PDL1 low expressed PDX models. And most importantly, our humanized PDX model phenotypically copied clinical anti-PD1 therapy resistant patients. You will see after the PD1 treatment in humanized mouse model, those cancer grow much quicker than the control group. In clinical, it's more like hyperprogressive disease. Yeah. We know Claudin 18.2, the subtype of Claudin 18, one like 4-pass transmembrane protein. It was only limitedly expressed in differentiated, like healthy epithelial cells of the gastric mucosa. However, it is significantly upregulated in a variety of malignant tumor tissues, such as gastric cancer, pancreatic cancer, and ovarian cancer. You will see here the growth of HEC293 Claudin 18.2 cells was gradually suppressed with the increased dose of anti-Claudin 18.2 antibody in our human PBMC and cancer co-inoculated model. And in our biobank, there are quite a few cases of Claudin 18.2 highly expressed gastric PDX samples. We also validate this data in IHC staining, so which could be used in various IO drug evaluation. We know dendritic cells are most powerful professional antigen presenting cells for inducing human-naive T cell activation and effective T cell differentiation. So currently, cancer vaccine and immune moderate targeting dendritic cells are still very hot. Here, we developed a monocyte-derived dendritic cell, cancer priming PBMC, and a fresh cancer co-transfer NCG mice model for anti-CD40 antibody in vivo test, and proved that the combination of anti-CD40 and PDL1 antibody fully stopped melanoma cancer development. And recently, CD47 SPA-alpha pathway was identified as a key innate immune checkpoint. Upregulate CD47 protein in cancer cell line is not only important for its intrinsic signaling for cancer itself, like proliferation and migration, but it's also important for cancer for the immune escape through its trans receptor like SPA-alpha on macrophage side by recruiting phosphase SHIP1 and SHIP2 for macrophage phagocytosis inhibition. And some clinical samples from triple-negative breast cancer patients. Those patients with higher level of CD47 and CD68 expression usually have very worse disease-free survival. Here we develop a monocyte-derived macrophage, M2 macrophage, mixed with tumor priming PBMC and fresh cancer cells, co-transfer into the NCG mouse. During this model, we will see anti-CD40 could synergize with another antibody for control the tumor glow. We also test anti-CD40 function in human B-cell leukemia cell line, NAM6, and some multiple myeloma patients, like PDX models, as well as some acute myeloid leukemia, also topically PDX models. Those models we use like mouse host immune system. Yeah. Parallel to in vivo, like anti-CD40 function study, we also run some in vitro, like antibody-dependent cellular phagocytosis, ADCP assays, which can help indicates that different anti-CD40 antibodies showed various cellular phagocytosis effect towards different human samples. Although their binding ability was very robust and clear, particularly for the blue dot line donor, the antibody showed no phagocytosis effect. However, the binding is quite nice compared with the IgG group.
That's great. Thanks a lot, Ben. And I'm sorry to do this to you again and cut in, but just another quick question here. So you've been talking about T cells and dendritic cells and also macrophages for the in vivo functional tests, but I know some drugs target NK cells. Do you have any models for those?
Sure. Yeah. We have already have some in vivo assays just by using fresh PBMC humanized models. As I showed previously for antibodies targeted Claudin 18.2, we actually run a fresh PBMC humanized model. So we proved its nice in vivo function. This antibody actually have an enhanced ADCC effect. It should mainly target NK cells. Besides, currently we also use some ex vivo expanded human NK cells and transfer into human interleukin-15 overexpressed transgenic mice model. So hopefully we can get some data soon. Yeah, thanks.
That's a great answer. Thanks again, Ben.
So next, I will briefly introduce our humanized models with effective immune cells. So besides PBMC, we also used novel CAR T cell for tumor curing in solid cancer. Here you will see some CAR T cell really did a great job. And here is the imaging for the tumor size. And also we have some data come from CAR NK cells plus interleukin-15 therapy. Although it seems the in vivo efficacy of CAR NK was not as efficient as CAR T cells, but it did well actually. We also have some data come from oncolytic virus, which could efficiently kill tumor cells by boosting immune system, such as overexpression like IL-12, IL-15, and IL-15 receptor, and some anti-PD-1 antibodies in the virus like genome. For Claudin 18.2 target, it is so interesting that not only its monoclonal antibody or bispecific antibody efficiently irradiate cancer cells, as I previously mentioned, but its antibody drug conjugates ADC drugs nicely suppressed multiple gastric cancer growth. We also run some in vivo antibody-dependent cell cytotoxicity ADCC assays to evaluate EC50 of antibody cytotoxic activities and do multiple cytokine detection by ELISA, bead-based Luminex, or flow cytometry for backtracking CAR T cell or oncolytic virus treatment-induced cytokine release syndrome in vivo. So next, I will briefly introduce the imaging mass cytometry, IMC, for mechanism or biomarker discovery in immunotherapy. In order to better decipher the mechanism of IO drug function in vivo, we took advantage of imaging mass cytometry, which composed a tissue imaging haplin and a detection site of system helos. as we know that mass cytometry used antibodies labeled with heavy metal ion tag rather than fluorochrome to avoid the compensation issue in classic flow cytometry. As the atom mass of each heavy metal is so unique, the peak is so sharp, there is no signal overlap for the detection. And similarly to the immunohistochemistry staining, IMC starts with a tissue section stained with multiple heavy metal labeled antibodies before loading into heparin and quantified by halos. IMC is such a powerful tool that it allows us to gain an appreciation of the law of heterogeneity in tissue spatial function and assess complex of phenotypes and tumor immune interactions in tumor microenvironment. For the early study of in vivo CAR-T infiltration in tumor tissue were persist with tumor digesting and staining with normal fluorescent antibodies for flow cytometry, and here is flow data. However, this method can't tell us the spatial interaction between cancer and immune cells. So by using IMAC analysis, we can easily locate there are some tissue, lymph node tissue in the lung cancer samples, like CD4 positive blue cells actually close to the red CD20 positive germinal center-like B cells, and some green CD8 positive cells. They surrounded by vitamin positive cancer-like epithelial cells, and those CD8 positive T cells also are granzyme positive as well. So IMC give us a lot of information about like the tissue location and spatial interaction between cancer and immune cells. IMC can also run like phenograph clusters, like transcriptome analysis and some class mapping to tissue image like 2D or 3D. Use this strategy currently, we are trying to help clinical physicians to identify which cell subset is sensitive or resistant to anti-PD-1 therapy. Now we have up to like 29 markers for the cyto and up to like 47 IMC panels ready to go, cover the main immune subsets, squamous cells, cancer cells, and the key signaling and the transcriptional factors, such as B7H3 and FOXP3 and CTLA4, PD1 and PDL1. Finally, with the fast development of immunotherapy, we have built a stable and well-tested humanized mice model platform for IO drugs, from in vivo to in vitro, from conventional like flow cytometry to tissue imaging mass cytometry. We started from clinical samples managed by a PMED trial database, followed by key cell sequencing based on 1,000 or 2,000 cells. Primary tumor conditional reprogram cell construction, and make some humanized patient-derived xenograft PDX model for various cancer drug evaluation. Thank you so much. Yeah.
Thanks, Dr. Ji. That was a fantastic presentation, and we'll now move right on to the Q&A. And our first question is it possible to use customized antibodies instead of the standard panel antibodies, since more and more innovative targets have been identified recently?
Oh, this is a nice question. Yes, definitely. We can do this. Since there's a lot of commercial available, like heavy mental labeled antibodies, like fluidigen, and also we can help to our customers to conjugate those antibodies in our facility. So we can do this since a lot of people they are interested in different parts of immune subtypes. Like some people focus on Th17 differentiation, so they probably want to label like our gamma T transcript factors, and some people may focus on cancer stem cell, so they prefer like EpCAM or like CD133. And most recently, a lot like novel cancer regulates has been discovered. So like a CD93 or CGLEC15, or just like today, I mean, in the latest journal of Cell, there's one interesting candidate called folate receptor beta, just published today. So it's for another new kind of macrophage subtype. It's so unique, contact with T cells. I believe most of the people wanted to conjugate and use them for their clinical or like translational work. Yeah, thanks.
Wonderful. We have another question for you. How do you determine the ratio of immune versus cancer cells in your co-injection models?
Oh, yes. If you go back to the slide I mentioned in the beginning, we did during our setting up this model, we do a different ratio, like a different ratio. So like from like 10:1 to probably to 2:1, we will find a different donor and between like PBMC and cancer cells to find an optimal like ratio to help to decide which is good for this model, because different cancers they maybe prefer different like optimal immune like sensing or interaction. So we have a particular like donor selection and a ratio testing before our real experiment. Based on this, we will have some like flow cytometry check, as I showed today before, and also we can do some fast imaging, like immunofluorescent image to quickly to match like day 1 after transfer, and we will check day 7 or day 14 or 3 weeks later. So the data I showed, one is during the day when we transfer from the imaging, and another is the flow cytometry check we did at like around like 3 weeks later. Yes.
Excellent, thank you. We have another question. One of the slides listed 12 targets that are already available for use on your platform. Are you planning on expanding those, or will it just be customized studies beyond those 12?
Which panel? You mean for which kind? For the classic flow, or for the for some other like a mass cytometry?
I believe that's for the classic flow.
Okay, so you mean for the classic flow, based on, because currently in our facility we have 3 laser and 11 color. So the maximum for our like conventional multicolor like a flow cytometry, we can run up to 11 channels. So that's why, I mean, for the conventional testing, if you wanted to decipher the immune cell phenotype, like hide or embed inside of the tumor, it's really hard. The maximum may be limited by the lasers. You can up to like BD, like for Tesla, you can run up to like 4 laser and 16 channels. And even more recently, if you use a spectrum flow cytometry like Cytec, it maybe can also up to like more than 25 based on different like spectrum like scanning. Anyway, those ones, they all have quite limited like numbers compared with like Satov, like mass cytometry. So I mean, another issue is composition issue. Yes.
Okay. Another question for you. Is your PBMC humanized mouse model stable, and how is the reproducibility of the assay system?
Oh, you mean for the co-transfer or for the like humanized models, and for first like a tumor bearing and transfer to a humanized model, like
for the
implant
humanized.
the separate. Okay, for the humanized model, we also have a donor selection first, because you know different donors give different rights to like T cells survival, or particularly in the immune system deficient mouse. So we have a donor selection to see whether they have a proper like ratio, not too high. If they grow too fast, I mean, so the mouse will develop to GVHD very, very faster. If they grow too low, maybe undetectable or less than 0.5 percentage, even after like 10 days after transfer, then we will abandon this donor. So usually we will double check whether it's like have a proper ratio. Then we can move to the next step, whether it is good. Usually we can run on the NCG mouse maybe like around like 5 to like 20, like when we check like at 3 weeks. So based on this, we can set up our very stable the humanized model like system to evaluate our IO drugs. Yeah.
Wonderful. And
quite repeatable.
Do you have labs in the United States? Oh, good. Do you have labs in the United States that can perform these studies?
Oh, currently, I believe we have marketing and a BD group or the some at least 2 units, I think, in US. The mainly experiment we well did in Shanghai in China, but we plan to have one laboratory maybe. If we have much more EU customer and US customers, probably we will set up one in Boston or in California. Yeah, based on the customers' request. Yeah.
Excellent. Okay, and then another question for you. What are the humanization percentages that you usually get?
Yeah, just like we usually focus on the CD45 positive, human CD45 positive, and the CD3 positive ratio. Based we double check. We will exclude the mouse T cells or mouse blood cells. So as I said, different model give different percentage, but usually if you want to set up a proper percentage and not develop too fast GVHD, then probably around like more than 5, but it's better less than like 20% out of the full like total live mouse blood pool cells. So it means no more than 1 out of 5, 1 out of 20% of the full like mouse blood cells, no more. Yeah.
Thank you, Ben, for sharing your insights. It's been a pleasure having you with us. And a big thanks also to the audience here for participating. Last but not least, we'd like to thank the sponsor, LIDE Biotech. And in closing, we hope you've enjoyed this Scientist.com webinar, and we'll see you again next time. Have a great rest of your day, everyone.